Hello, this is Seher from Easy Peasy, and the topic we are going to discuss today is called as transport of protein into mitochondria. As we know that mitochondria is the powerhouse of the cell, as it provides energy to each organelle in the form of ATP molecules. So let's take a closer look at the structure of mitochondria. If we look closely, we can see that mitochondria consist of two membranes. The outer membrane is rounded in shape, as you can see in this picture in the red color. An inner membrane is in yellow color and it is not rounded, rather they are making a lot of different kinds of folding that is called as cristae. The space that is present in between the outer membrane and the inner membrane is called as intermembrane space. And matrix is the inner portion of the mitochondria visible in blue color. They also have granules and ATP synthase particles because the major function of mitochondria is to produce ATP. Mitochondria also contains ribosome and deoxyribonucleic acid. It means that mitochondria can produce its own protein. But if we look at the biogenesis of the mitochondrial protein, we can see that only 1% of the mitochondrial proteins are generated inside the mitochondria, and 99% of the proteins are coming from the cytosol and then they are imported inside the mitochondria. The function of mitochondrial protein is the energy metabolism, metabolism of amino acids, lipid, heme, and iron, transporters of metabolites, protein transport and folding, protein degradation, signaling processes, membrane remodeling, fusion and fission, involvement in programmed cell death. Now let's take a closer look at the membrane structure of mitochondria to understand that how the proteins are importing inside the mitochondria. So this is the mitochondrial part that we are going to elaborate now. This is the outer membrane of mitochondria and this is the inner membrane of mitochondria. It is not straight because they make a lot of different foldings called cristae. Mitochondria usually attach itself with the endoplasmic reticulum by a domain called as ERMES. The full form of ERMES is endoplasmic reticulum mitochondria encounter structure. This ERMES subunits are really important because they are required for the maintenance of mitochondrial morphology, mitochondrial genome maintenance, and assembly of the outer membrane protein. The outer mitochondrial membrane is also connected with the inner membrane by a structure called as MICOS or MICOS. The full form of MICOS is mitochondrial contact site and cristae organizing system. Now, MICOS are required for the maintenance of typical mitochondrial ultrastructure to facilitate the formation of membrane contact site and facilitate the formation of crystal junctions. Okay. Now we have a lot of different type of proteins that are present on the outer membrane and the inner membrane of the mitochondria. And these proteins collectively are called as translocons. With the help of these translocons, the protein enters the mitochondrial membrane and reaches its destination port. The process of importing protein into the mitochondrial membrane consists of 4 steps. First step is the synthesis of that protein. So for that, we need a ribosome, messenger RNA, and protein. So this red color is basically showing that protein is coming out of this translational machinery. The second step is the recognition. Recognition of targeting signals by targeting factors. If this protein needs to target mitochondria, so the targeting signal will be called as mitochondrial import sequence. And the targeting factors that are going to attach itself with this protein is called as HSP70 and HSP90. They are basically the chaperones or heat shock proteins that are going to attach itself with the protein and will not allow it to transform into tertiary and quaternary structure. The third step is called as targeting. Targeting of this protein to the receptor present on that specific membrane. This receptor is the translocon present on the outer membrane of the mitochondria. The fourth step is called as translocation or insertion of the protein. Once the protein enters the outer membrane, then they will be targeted to the subcompartment of that mitochondria. By this way, we can recognize 4 subcompartments where a protein can target itself. The first destination is the outer membrane. The second destination is the intermembrane space. The third can be inner membrane, and the fourth can be matrix. In this picture, we can see that protein have 2 different types of signals as well. One is called as N-terminal pre-sequence, which have these positively charged amino acids on it, and one is called as internal targeting signal. To elaborate the signals present on mitochondria, we can see this table. Now, to understand this table, let's start from the right side. Now, this is the pre-sequence variation. All the pre-sequence proteins targeted itself to the matrix, inner membrane, and intermembrane space. And all of these proteins are using the same type of translocons to reach their destination. Now, if the protein is targeting the matrix, then they will have the empiphathic structure on the N-terminal side of that protein, and it will be cleaved once it reaches the matrix by the enzyme called as mitochondrial processing peptidase. If the protein is targeting the inner membrane, then they need a hydrophobic part to integrate in that membrane structure. For that, as you can see in this picture, that they have the pre-sequence plus a hydrophobic region shown in the yellow box here. And this pre-sequence will be detached once it reaches to its inner membrane destination. The third type of sequence is going to stay in the intermembrane space. Now, in the intermembrane space, they don't need the hydrophobic region as they are not going to integrate this protein into any membrane. So, if they do have this hydrophobic region, as you can see in this picture, it will be cleaved by inner membrane proteases. The second category we have is called as non-cleavable signals of hydrophobic protein. Now, if the protein doesn't have the pre-sequences, then there is a 90% chance that they are going to integrate themselves within a membrane. As you can see in this table, all these destinations are in the outer membrane, inner membrane, and inner membrane. They are using different type of translocons that we are going to discuss in this video. So let's discuss the first signal, that is the beta signal or beta barrel signal. Now, this protein have 2 signals. One is called as Tom signal and one is called as beta signal. The beta signal is present near the C terminal of this protein. When the protein comes out, it is going to enter the outer membrane first by Tom signal and then integrate it in the outer membrane by the beta signal. The second structure is called as the alpha helical proteins. Now, they have 3 variations. The first variation is that the sequence is present near the N-terminal that is going to integrate this protein in the membrane, and it is called as signal anchored sequence. It can also be present near the C-terminal, so it will be called as C-tail anchor. Or it can present in the center of this protein and will be called as internal signal. Some proteins have multi-spanning, so it will cross the membrane more than once. For that, we have various hydrophobic regions present on that protein, and this green portion will be used to integrate this protein in the membrane. So this protein is going to enter the membrane 3 times like this. Pre-sequence like internal sequence after hydrophobic region. Now, this type of protein is usually target the inner membrane of that mitochondria, and it has a hydrophobic region plus the empiphathic molecule. This hydrophobic region is used to integrate this protein in the inner membrane, and this pre-sequence-like structure will hang in the matrix. The third type of signal is called as internal signal for intermembrane space. Now, this protein stays in the intermembrane space. They used a translocon called MIA that we will discuss in detail in this video, and this protein usually have cysteine molecules to make disulfide bonds. Now, let's discuss each process in detail. So this is the outer membrane and inner membrane of the mitochondria. And the first translocon that we are going to discuss here is called as Tim complex. Tim complex is the major protein that is present on the outer membrane of the mitochondria. The full form of Tim is called as translocase of the outer mitochondrial membrane. Now let's see the structure of this Tim complex. Tim complex consists of central pore region called as Tim40. They have Tim22 that is called as central receptor. Tim70 and Tim20 is called as peripheral receptors. Tim5 assists with the transfer of precursor proteins. Tim6 stabilizes the Tim complex, and Tim7 usually assists the Tim dissociation. Now let's see how a protein enters from the Tim complex. Now, Tim complex is used to insert the protein from cytosol into the intermembrane space of mitochondria. So this is the protein that needs to enter this Tim complex, and this red portion is basically the sequence that is going to tell that this protein needs to enter the mitochondria. Now, this protein will be attached by chaperones that are most likely called as HSP70 and HSP90. These chaperones will not allow this protein to turn from primary stage to tertiary or quaternary stage. Now, the pre-sequence usually get recognized by Tim20, and the chaperones are recognized by co-chaperones that are mostly present on Tim22 and Tim70. Now, for example, in this picture, the co-chaperones that are going to recognize the chaperones on protein is called as XDJ1 and DJP1. Now, they will recognize these chaperones, and pre-sequence will be recognized by Tim20. By this way, they will let this protein to enter the core complex by Tim40 like this. Once the protein enters inside the intermembrane space of mitochondria, the pre-sequence will be cleaved by inner membrane proteases like this. Now, if this protein's destination is the intermembrane space, then it is allowed to transfer into tertiary or quaternary stage like this. If not, then it will remain in the primary stage and will transfer to its destination by using other translocons that we will discuss in this video. Now let's see how this protein will convert itself into tertiary and quaternary stage. Now this is the protein that needs to turn into the tertiary and quaternary stage. For that, this protein needs to have cysteine residues in it. To maintain the primary stage of this protein, small Tim complexes called as Tim12, Tim9, Tim10 will attach themselves to it. Now, this protein needs a translocon that will help it to turn into its functional tertiary or quaternary stage. For that, the translocon that is going to help it is called as MIA complex. The full form of MIA complex is called as Mitochondrial Intermembrane Space Assembly Machinery. MIA complex have two domains. One is called as MIA40 and one is called as ERV1. Now, MIA40 have the properties of oxidoreductase and ERV1 have the properties of sulfhydryl oxidase. Now let's see how MIA complex is going to help this protein to create disulfide bonds between them and turn into its functional state. Now this is a beautiful picture showing the whole process how MIA complex is working. Now this is the precursor protein that needs to transfer inside the intermembrane space. As you can see in this protein, we have a lot of cysteine residues that is represented by SH. Now this protein is going to pass through the Tim complex. When it comes out of the Tim complex, MIA40 will make bond with the sulfur atom present on cysteine amino acid. So MIA40 will recognize this protein initially. Once the recognition is done, MIA40 will recruit ERV1. ERV1 is going to oxidize this protein by making disulfide bonds between them. Now, once the protein will take its functional stage, the tiny Tim complexes will remove itself from it. Now, the ERV1 and MIA complex will detach from itself, and ERV1 is going to reoxidize by transferring the electrons to the respiratory chain and will be available for the next protein that needs to stay inside the intermembrane space. The next translocon that we are going to talk about is called as MIM complex. The full form of MIM complex is mitochondrial import complex. The function of this complex is that they will take alpha helical proteins and they will import these proteins inside the outer membrane of the mitochondria. The second way to incorporate this alpha helical structure inside the outer membrane is that this protein will enter the intermembrane space through Tom complex, and then they will take the help of MIM complex and then integrate itself inside the outer membrane of the mitochondria. Now, this alpha helical structure can be of single spanning or can be of multi-spanning. Now, we have 4 different types of alpha helical structures that can incorporate in the outer membrane of the mitochondria. The first sequence is called as end-signal anchor sequence. As you can see that the hydrophobic region is present near the N-terminal, and it is used to incorporate this protein inside the membrane, and the rest of the protein will stay inside the cytosol. The second type of sequence is called a C-tail anchored sequence. As you can see in this type of sequence, the hydrophobic region is present near the C-terminal, and the N-terminal region of the protein will hang in the cytosol. Internal signal means that the hydrophobic region of the protein will be present in the center of that protein. So half of the protein will be hanging in the cytosol and half of the protein will be hanging in the intermembrane space of mitochondria. And the last is called as multi-transmembrane segments, as it can penetrate the membrane several times due to the presence of several hydrophobic regions inside this protein. Now, MIM complex can incorporate these alpha helical structures inside the membrane by 3 different ways. The first way is this MIM complex will take the help of Tom70 and incorporate the protein inside the outer membrane. In some situations, MIM complex can incorporate the protein in the outer membrane independently. And sometimes the MIM complex take the help of SAM complex and incorporate the protein inside the outer membrane. One of the studies shows that signal anchor proteins take the help of Tom20 and Tom70 proteins, and tail anchor proteins will take help of Tom5, Tom6, and Tom7 proteins. And these are the real-life examples of the proteins that enter inside the membrane. These proteins are multi-spanning, and these proteins are entering from the intermembrane space of the mitochondria. The next translocon that we are going to discuss right now is the SAM complex. SAM stands for Sorting and Assembly Machinery. The function of SAM complex is to incorporate beta barrel protein from intermembrane space into the outer membrane of the mitochondria. SAM complex consists of 3 domains. SAM50 is the core domain from which the beta barrel protein will assemble itself and then incorporate it into the outer membrane. SAM35 and SAM37 provide stability to the SAM complex, and SAM50 have a domain that is going inside the intermembrane space called as POTRA. This POTRA is basically the receptor domain that will recognize the protein from intermembrane space, and then it will incorporate it into the outer membrane. The full form of POTRA is Polypeptide Transport Associated Domain. Now let's see how it's going to perform its function. First of all, the protein from the cytosol will pass through the Tom complex, because as we know from the table, beta barrel proteins have 2 domain sequences inside that protein. The first sequence is called as Tom signal. It means that it will pass through the Tom complex first, and then it has the beta signal near the C terminal, and then it will use the SAM complex and will incorporate itself in the outer membrane in the form of a beta barrel. So as you can see in this picture, this yellowish portion is basically this protein. It is passing through the Tim complex into the intermembrane space. Here, in order to avoid this protein to turn back into the tertiary and quaternary state, Tim9 and Tim10 will help it and will not allow it to fold back on itself. Then it will shift itself into the SAM complex, and SAM complex will shift this protein in the form of a beta barrel in the outer membrane of mitochondria. This is basically the assembly part. As you can see that the protein is using the partner domain and the core complex in order to assemble itself and then move itself outwards in the outer membrane. The secondary function of the SAM complex is the SAM complex is basically playing an important role in making the ER-MES complex and MICOS complex. So it is basically connecting the inner membrane, the outer membrane, and endoplasmic reticulum together on the mitochondria. So it is playing a big part in the stability of the mitochondria. The next translocon that we are going to talk about is called as Tim complex. Now we have two different types of Tim complex. One is called as Tim23 and one is called as Tim22. They both perform different functions, so we will discuss them one by one. So right now we are going to discuss the structure of Tim23. The full form of Tim complex is translocase of the inner mitochondrial membrane. Tim50 and Tim21 is basically the receptor domain that will recognize the pre-sequence coming from the Tim. Tim23 and Tim17 is going to play a role of core domain and allow the protein to pass through it from intermembrane space into the matrix. Tim23 is connected with a PAM complex. The full form of PAM is pre-sequence translocase associated import motor. SAM complex is playing a role of pulling the protein into the matrix. It is connected with the chaperone, that is mitochondrial HSP70, which is using the ATP molecule in order to pull the protein from intermembrane space into the matrix. Then we have MGE1, that is a co-chaperone that is stabilizing the attachment of HSP70 with the PAM complex. We also have another co-chaperone called as ZIM17 that is preventing the HSP70 from aggregation. Now let's see how this Tim23 is going to perform its function. Now this is a step-by-step picture for how the Tim23 is going to work. Right now there is no protein coming up. This is the outer membrane having the Tim complex, and this is the inner membrane having the Tim23 complex. Right now the PAM complex is not attached to it and the Tim23 is closed because Tim21 and 50 are just connected with each other. When the protein will be recognized and pass through the Tim complex, the pre-sequence of that protein will be recognized by Tim50 and Tim21. By this recognition, Tim50 and Tim21 will move apart and open the Tim23 core. Now to pull this protein into the Tim complex, the membrane potential will develop by this sign, and it helps the protein to move from the Tim complex. Now some more proteins will come and attach themselves with this Tim complex, that is COX and BC1. COX is basically the cytochrome C oxidase enzyme, and BC1 is the respiratory chain complexes 3. With the attachment of these proteins, we have two options now. If this protein destination is the inner membrane, then we have this protease called as mitochondrial processing peptidase. This protein will remove the pre-sequence part of this protein and will allow it to integrate itself into the inner membrane. If not, then PAM complex will come and attach itself with the Tim complex. PAM complex recruit HSP70, and HSP70 is a chaperone that will use ATP molecules and will start pulling this protein from the intermembrane space into the matrix. Once all the protein is inside the matrix, this mitochondrial processing peptidase will cut the pre-sequence part and will leave the protein. Now let's see what happened to this protein when it is inside the matrix. Now this is the same type of thing that is happening in the previous picture. This is the Tom complex, this is Tim23 complex, and the protein is inserted by the chaperone HSP70 by using the ATP molecule. Now once the protein is inside the matrix, some of the more enzymes will come and will modify this protein. The first enzyme that will come and remove the N-terminal amino acid from this protein is called as intermediate cleaving peptidase. And the second enzyme that's going to come and remove some of the nucleotides from the N-terminal, and that is called as octapeptidyl aminopeptidase. Now this protein will move towards another chaperone that is called as HSP60. With the help of HSP10, they will help the protein to fold back into its functional form. The next translocon that we are going to talk about right now is called as Tim22. The full form of Tim complex is translocase of the inner mitochondrial membrane. Now Tim22 have the Tim complex that is a core domain, and it has Tim54 and Tim18 that will act as a receptor domain and will recognize the protein from the intermembrane space. Now let's see how it's going to perform its function. Now here, as you can see, this is the outer membrane, this is the inner membrane, and this is the cytosol. Now protein from the cytosol is attached itself with the chaperones that are HSP70 or HSP90 and will recruit inside the intermembrane space from the Tom complex. Once the protein is inside the intermembrane space, Tim9 and Tim10 will attach themselves with it and will not allow it to fold back on itself. Now this protein will be recognized by Tim54 and Tim18 and will put this protein in the core domain of Tim22. From here, the Tim22 complex will insert this protein in the inner membrane. Last but not least, the translocon that we are going to talk about is called as Auxar Complex. Now Auxar stands for Oxidase Assembly Machinery. The function of Auxar Complex is to incorporate the protein from the matrix into the inner membrane. As we know that mitochondria have mitochondrial DNA present in the matrix, and it also has some ribosomes, so it can produce 1% of the protein inside the matrix. This protein, if its destination is the inner membrane, can use this Auxar complex and will integrate itself into the inner membrane. Now this is a good picture showing us that how the protein is moving from mitochondrial matrix to the inner membrane. A protein from the cytosol can integrate itself into the inner membrane after penetrating to the matrix. As you can see in this picture, this is the protein recognized by the Tom complex, then moving in the Tim complex. The HSP70 is pulling it inside the matrix. Now here, the protease is going to cleave the pre-sequence, and this protein, if it wants to go back into the inner membrane, can take help of this Auxar complex and will integrate itself into the inner membrane. Auxar complex is also working on the other side. Now this is the ribosome inside the matrix, and the protein is coming out of it. While it is still attached itself with the ribosome, the protein's first part will attach itself with the Auxar1. And Auxar1, with the help of MBA1, will start integrating this protein into the inner membrane. Thank you very much for watching this video. If you like it, please subscribe this channel. Thank you. Bye-bye.